All right. This is still chapter 17, and this is part 2, and it's on erythrocytes, or red blood cells. So, red blood cells are small in diameter, about 7.5 micrometers, so really, really tiny, and they contribute to gas transport. They have this biconcave dish shape, which you can see here. So you can see it's got this concave part on both sides. And what this is going to do is this is going to help it transport oxygen. The erythrocyte is anucleate, has essentially no organelles. It's filled with hemoglobin for gas transport, which is great. And red blood cell diameters are larger than some capillaries. And then the plasma membrane has this protein called spectrin, as well as some other proteins. But what spectrin does is it provides flexibility to change shape. Now, in AMP1, we talked a lot about how we study anatomy and physiology together because structure and function are complementary of each other. And a red blood cell is an awesome, superb example of complementary structure and function. There are 3 things that make it efficient for gas transport. That biconcave shape that I mentioned a moment ago, it gives it a huge surface area relative to volume for gas exchange. Hemoglobin makes up about 97% of the cell volume, not counting water. And then red blood cells have no mitochondria. And you remember, mitochondria are what produce ATP. So ATP production in red blood cells is going to be anaerobic, so they do not consume the oxygen they transport. How awesome is that? So even though they have an abundance of oxygen that they could be using, they do not use it at all. And again, this is what an erythrocyte looks like. And then the function of erythrocytes. So they are dedicated to respiratory gas exchange. And then this is where we really start talking about hemoglobin. And hemoglobin binds reversibly with oxygen. In males, the normal values are going to be somewhere between 13 to 18 grams per 100 milliliter. And then in females, it's going to be somewhere between 12 to 16 grams per 100 milliliter. Hemoglobin consists of a red heme pigment bound to the protein globin. And then globin is composed of 4, and that's important, 4 polypeptide chains, 2 alpha and 2 beta. And then the heme pigment is going to be bonded to each globin chain, and it's actually going to give blood its red color. And then each heme's central iron atom binds 1 oxygen. So we have the 4 chains, 4 globin chains, beta, beta, alpha, whoops, sorry about that, alpha, and then a heme associated with each one. And then the oxygen is going to bind to the iron atom in the middle of the heme. This means that each hemoglobin molecule is going to be able to transport 4 oxygen molecules. And then each red blood cell contains 250 million hemoglobin molecules. That's a lot of hemoglobin. So O2 loading in lungs is going to produce oxyhemoglobin, which is a ruby red color. And then oxygen unloading in tissues is going to produce deoxyhemoglobin, which is a dark red color. And then sometimes, or about 20% of the CO2 in the blood will bind to hemoglobin, and that's going to produce something called carbaminohemoglobin. And then we start talking about how erythrocytes are produced. So first, we introduced the term hematopoiesis, and this is one that you probably came across in AMP1. And hematopoiesis is the formation of all blood cells. And this is going to occur in red bone marrow. This is composed of reticular connective tissue and blood sinusoids. In the adult, you're going to find red bone marrow in the axial skeleton, so the skull, the thoracic cage, the vertebra, the girdles, particularly the pelvis, and then the proximal epiphyses of the humerus and the femur. And remember, the proximal epiphyses are the ends of the long bones. Well, not their proximal epiphyses, the epiphyses, are the ends of long bones, and the proximal epiphyses are going to be the ends of the bones that are attached to the trunk. Hematopoietic stem cells, or hemocytoblasts, are stem cells that give rise to all formed elements. And then we have 2 things that kind of push cells towards a specific pathway of blood cell development, and that's hormones and growth factors. And then once the cell is committed, it cannot change. And then new blood cells are going to enter blood sinusoids. So we have a regulation and requirements of erythropoiesis. So too few red blood cells can lead to tissue hypoxia. That's low oxygen levels. And too many can increase blood viscosity. And that's because you're putting more formed elements in a fluid, and that's literally just going to make it's going to increase the viscosity, which is going to put possibly a strain on the heart. More than 2 million red blood cells are made per second, and the balance between red blood cell production and destruction depends on two things. The first one that we'll talk about are hormonal controls, and the second one we'll talk about are dietary requirements. So for hormonal control, we talk about EPO, or erythropoietin. And EPO is a hormone that stimulates the formation of red blood cells. There's always a small amount of EPO in the blood to maintain basal rate. And kidneys are going to be the organ, and then to a small degree the liver, that's going to release EPO, and it's going to be released in response to hypoxia. So specifically getting down to under what circumstances it would be released, we have under circumstances where there's low oxygen levels. There are oxygen-sensitive enzymes in the kidney cells that cannot degrade something called hypoxia-inducible factor. Hypoxia-inducible factor will accumulate, and that accumulation will then trigger the synthesis of EPO. Some of the causes of hypoxia could be decreased numbers of red blood cells due to hemorrhage, like a wound or a bleeding ulcer, or increased destruction. Let's say maybe somebody has sickle cell anemia. Insufficient hemoglobin per red blood cell, like an iron deficiency, or reduced availability of oxygen. So high altitudes or lung problems such as pneumonia, or even you could say COVID. Too many erythrocytes or high oxygen levels in the blood inhibit EPO production. EPO causes erythrocytes to mature faster, and then testosterone enhances EPO production, which is going to result in higher red blood cell counts in males. So if we were to look at this, we have this balancing act, right? So we have hypoxia, which is inadequate oxygen delivery due to a number of things: a decrease in red blood cell count, decrease in hemoglobin, decrease in O2 availability. And this is going to stimulate the kidney, and then to a lesser extent the liver, to release EPO. EPO stimulates red bone marrow. This is going to enhance erythropoiesis, which is going to increase red blood cell count, which is then going to increase the oxygen carrying capacity of the blood. The next thing we talk about with regulation and requirements of erythropoiesis are the dietary requirements. And these are amino acids, lipids, carbohydrates. And then we also need iron, really important, right? Because we find iron in hemoglobin. And iron is available from the diet. 65% is found in hemoglobin. The rest is found in the liver, spleen, bone marrow. Now, free iron ions are toxic, so they are going to be bound to proteins. They can be stored as ferritin or hemosiderin, and then transported in the blood bound to transferrin. And then vitamin B12 and folic acid are necessary for DNA synthesis for rapidly dividing cells such as red blood cells. So red blood cells only last about 100 to 120 days. They are anucleate, so they don't synthesize new proteins, they don't grow, they don't divide. When they become old, they become fragile, the hemoglobin begins to degenerate. They get trapped in smaller circulatory channels, especially the spleen, and that is where we will find macrophages that will engulf and break down dying red blood cells. Now, as a red blood cell breaks down, the heme, the iron, and the globin are separated. The iron is going to bind to ferritin or hemosiderin and is stored for reuse. Heme is going to be degraded to the yellow pigment bilirubin. The liver is going to secrete bilirubin into the intestines, where it is degraded into the pigment urobilinogen. Urobilinogen is then transformed into the brown pigment stercobilin, which leaves the body in feces. And then globin is just metabolized into amino acids and released into circulation. These are the two questions for this particular section. I hope you guys have a good day.